spatial site frequencies.
Okay, thank you. Thank you everybody for coming today. I'm going to talk about a new summary statistic that I developed in collaboration with Mike Hickerson to infer historical spatial dynamics. And so I don't think I need to convince you that the species ranges are not constant through time. And the best examples we have are from introduced species, such as this little finch over here, which in a matter of decades expand really quickly from their point of introduction in northeastern North America. Of course, this is not a pattern that happens only at those short temporal scales. It is something that we know happens on a much greater time scales, usually associated with climate change. So for instance, in this example, what you see on the y-axis is the area of the geographic range of each of these four mammals. And the only point I want you to take from this is that the area that they occupy change over time. And if we think about this, actually, since the origin of the species, the niche is bound to change. The geographic range is bound to change, either through processes like dispersal, local extirpation, or long-distance jumps. And one of the features most commonly happening about range expansion is about changes in the range is range expansion. The best known example comes from our own history as a species. I think by now there is really little debate about our origin in Western Africa, and from there, through a series of founder events, we colonized the rest of the world to the current present distribution. So what I want to point out here is the key aspect of this model is this decrease in population size, represented here by a smaller width of these shapes, that later recovers to attain a bigger sample size. So for each time step, what we see is a founder or a bottleneck. The key thing is that we won't be able to understand the dynamics of range expansion and how it affects patterns of genetic diversity unless we really have a clear idea of where it originated. And so what I'm going to introduce here is this method that actually is meant to capture information on relative allele frequencies and their explicit spatial distribution of heterozygosity populations have as a product of these serial founder events. So they use that information to look at the origins of expansion. The way they did that is they basically feed this regression using as the source every single cell in the world. And then they come out with the idea that the one that has the greatest fit, the greatest R-square, is the region where the populations are expanding. They was applied for human data. You can see there is a fairly good match, although it's a little bit to the eastern of what we currently accept is the origin. More recently, Peter and Slatkin came with another approach that is called the directionality index. This approach basically is a comparison of allele frequencies between pairs of population. The underlying notion for this statistic is this common pattern of reduction in the number of alleles represented. Hopefully you can see over here there are circles of different colors, and the number of colors keep reducing as you move away from the source. What basically that shows is that some alleles will reach fixation, some other alleles will disappear from the population, and therefore you can capture those relative frequencies between population pairs to infer where the origin is. And they proved their method comparing two models, an isolation by distance and a serial founder expansion model. And as expected, they show that under an isolation by distance model, their index doesn't change with the distance from the source, whereas in a serial founder event, it actually keeps increasing as you move away from the source. Furthermore, they also show that previous methods used to identify expansion sources, such as the heterozygosity method that I just explained, and FSD, do a poorly good bad job under those models. There is an issue with this index, although it has been proven to work, and it has to do with the amount of data it needs. So the directionality index considers shared non-ancestral alleles only, which basically means part of your data are going to be thrown out no matter what. All the private alleles, you are not taking them into account. The other aspect of it is because you need to know the polarity of the SNPs, in this case, which SNPs are ancestral, which are derived, it only works for a species where you have that information, where you have that outgroup. However, we know that because of sampling issues and sometimes cost, we cannot get information about the outgroup for all the species we're interested in. The other issue with this index is that it heavily depends on the amount of data. In particular, what they found as the best performance requires 20 individuals per locality sample. And this could be cost prohibitive for many non-model organisms. So we came with this idea of trying to find a statistic that works in these more challenging circumstances. So one in which you don't have any outgroup, and basically we went away with that by using the minor allele as opposed to the derived allele. And the minor allele is nothing else than the allele that is at the lower frequency, at the lowest frequency in your sample. We developed also for much smaller samples, so we're only using one individual per locality. We're using all information, both shared and private alleles, and hopefully this is something we're working on. It can deal with more complicated spatial histories, such as multiple sources, as well as cyclical models, in which you have patterns of expansion and contraction repeated over time, which, as we know, happen during the Pleistocene cycles. So let me briefly explain how it works. It's a little bit complicated to do this in this short talk, but hopefully it will be enough. So let's imagine you have one site over here, a locality. So this is longitude, this is latitude. And one thing I want to point out is by site, I'm not referring to population in the genetic sense. And actually that's the beauty of our method. You don't need to assign individuals to population. All you need is the coordinates where you collected that particular individual. And so let's imagine in this particular case that from that side there is a range expansion. So from side 1 there is one that goes to the east, to side 4, and one to go to the west, side 3. And so let's sample individuals in those localities. I know this is a little bit complicated, so let me go through it. So on the columns, you have different SNPs. So in this particular case, I have up to 8 SNPs for this individual. And on the rows, I have the 2 alleles, assuming it's a diploid organism. So I have a maternal and a paternal allele. In color is presented those cases where there is a minor allele. So if you see here, it has the ancestral allele. In this position, it has a minor allele for that SNP. So one thing we can do is actually quantify the distances between all alleles or all homologous alleles. So let's go through an example here. So let's again consider site 1 and the individual there. And then what I'm going to do is look at the first SNP, the first locus you have. So you see, what the question you are asking basically is, where else can I find this allele? And in this particular example, if this works there, you can see that it only occurs in the individual in site 3, and that individual has 2 copies of it. So what I'm going to do is construct a distance vector that accounts for that distance. This is distance 1, there are 2 copies, and therefore I have 2 distances there. Now I move to the second allele in that particular SNP position. It's the same minor allele, so basically I just extend this twice. I do that for all the positions, for all sites, and what I end up, basically in a nutshell, is for each locus I will compile the spatial distances between copies of the minor allele. What that will give me is sort of this vector, which has 2 kinds of information. First, it accounts for pairwise relative frequency, some of the previous methods we're trying to do, but in addition to that, it accounts for the spatial explicit distribution of those alleles. So what I will do then is compile all that information into a single distance vector per site, per sampling locality. And I will pin those distances into a histogram using a Sturge's equation. Basically what that will give me is what we're calling the spatial-SFS. So far so good, we have the summary statistic. Now you're asking how we're going to go about inferring the source. Here's how we do it. One thing that you will notice is that the irregularity of that spatial-SFS has to do with the distance from which the population originated, with the most irregular occurring at the expansion source and the less irregular occurring farther away. So we can actually quantify this with a statistic, and I'm borrowing Harpening's ruggedness index, which he developed more than a decade ago under a different context, but for this, the purpose of quantifying irregularity works quite nicely here. So what you will have is an origin with a ruggedness index associated for each location, And what then you can do is use a spatial interpolation to identify what would be the ruggedness in portions of the landscape where you don't have samples. However, for simplicity for this talk, I'm not going to talk about that option, and I'm just going to work with the actual values without interpolation. To test the power of this method, what I did is a series of simulations, both in a 1D model, which basically I randomly choose a source from which the population expanded until the entire area was colonized. And I also have a 2D model where you see the same pattern. You identify a source from which the population expanded. In addition, I have a second 2D model where I have two sources to explore how the number of sources affect the power of the method. For the purpose of this talk, however, I'm just going to focus on the results of the one-source model. The parameters for these simulations: first, you have sample DIMs. I always sample randomly 10 DIMs, which are these blue dots. For each DIM, I set the population size at 1,000 individuals and the number of SNPs at 5,000. And I have 4 parameters that I vary with the simulations. Migration rate between DIMs, the strength of the founder event, which basically means how many new colonizers move out in each generation, the expansion rate, which means how fast the expansion happened, finally the time after the expansion ended, that is the time after all DIMs have been colonized, that I allow the simulation to keep running, basically in a model of isolation by distance. Here are the results. It's a little bit complicated to see, so let me first go over how to interpret the results. What you will see here is on the x-axis is the parameter of interest, in this case the time after expansion. On the y-axis, you see the root mean square error, which basically you can understand like the distance to the real source. If you are here at 0, like in this case, I'm doing really well, no matter the conditions. For the most part, I'm identifying the correct source. Up here, I have some issues. And how many of my simulations falls in each category depends on this color scale that you see here. In addition, to simplify the visualization, I'm going to present the same density in a univariate way on the side. And what you see here is the overall accuracy across the entire range of parameters, as well as the Pearson correlation coefficient, which basically identifies how the accuracy changes with the parameter value. In this case, being a negative coefficient, that means that I'm doing worse as the time after expansion increases, which is what you will expect because isolation by distance erases the pattern of the expansion. In addition, I'm going to compare those as a benchmark with the directionality index, but I want to point out that this is sort of an unfair comparison because, as I mentioned before, this method was not meant to deal with one individual and with the minor allele. So take it with a grain of salt. So here is the results you just saw, and here is basically for the 4 parameters. You can see that overall we're doing pretty well in identifying the source, with the only exception of migration, in which when migration is higher, I have a small power to identify when it's a 1D model. In all cases, there is a negative association with the parameter values, and the most stronger one is the founder strength. In the 2D model, we do much better, and basically the reason for that is the fact that I have a better spatial representation of genetic variation when you consider the 2 models. And you can see again, I'm doing really well no matter what, and still is correlated with the parameters themselves. So really quickly go over an empirical example where we apply this method. This is Lycaedes butterflies on Western North America, and what we did, we chose this study system because of the strong changes in vegetation and the glaciation extent impacted heavily this population. From the entire set, this is a data set by Comper 2004. I selected only 2 species, and we have, as you can see, over 15,000 SNPs. And what I did sort of to prove of concept is I created niche models for the last LGM, so I created them in the past and in the present and retroproject to the past and asked whether my method will identify correctly areas of high suitability, as you will expect, is where they actually stay. And I'm running out of time, but really quickly, you can see that in one case it does identify high suitable areas, seen here. In the other case, it doesn't. But when you look why that is, it's basically these are statistics that tell you how the model is performing. And as you can see here, this is a bad niche model, this is a good niche model, and when I have a good niche model, we do pretty well. And with that, I'm just going to point out that the spatial-SFS works, that it's even coming out pretty quickly, and that there are these three factors: the strength of founder event, the amount of migration, of the number of sources, the things you need to consider when applying this statistic. Thank you.